Dear participants, we are about to start the session. A warm good afternoon to the dignitaries and participants in Malaysia, and a pleasant good morning to dignitaries and participants in India. These two different greetings are due to the time difference between the Malaysia and India, and Malaysia standard time is 2 hours 30 minutes ahead to India standard time. Welcome to the international webinar on challenges and innovative drug delivery approaches for the management of chronic diseases. This is Dr. Ravi Shushala, the moderator of today's webinar. This is the first international webinar jointly organized by Faculty of Pharmacy, UiTM, University Technology, Mara, Malaysia, and the School of Pharmacy, GITAM deemed to be University, Hyderabad, India, under the MOU between both universities signed in the year 2019. Today's webinar is a virtual platform for exchange of ideas and expertise about the role of pharma and biotech professionals in developing novel strategies for the management of chronic diseases across the globe. And for this, today we are having two distinguished speakers for the webinar. Prof. Wang Tinwai from Faculty of Pharmacy, University Technology Mara, UiTM, Malaysia. I'm Dr. Sunita Sampati from GITAM deemed to be University, Hyderabad, India. To start the session, firstly I request Dr. Sherija, Dean, Faculty of Pharmacy, UiTM, Malaysia, for the opening remarks.
Thank you, Dr. Ravi. Hello, everyone. So, Honorable Prof. Dr. J. Shivakumar, Principal of School of Pharmacy, GITAM. Our respected speakers, Professor Dr. Wang Tinwai, Dr. Sunita Sampati, the organizing committee, my fellow colleagues, ladies and gentlemen. So, good afternoon. So, I'm sure in Hyderabad it's also afternoon, 12 o'clock. Okay, so good afternoon. It is my pleasure to welcome you here today to share with us a special moment. I think it's a mark of a new page of cooperation between a Faculty of Pharmacy, UiTM, and the School of Pharmacy, GITAM. So I strongly believe that our combined strengths in education as well as research expertise will take us to the next level. With us learning from each other, most important of all, I think we will build a strong and professional team, not just to cultivate our research talents together, but also to strengthen and diversify our education, execution, and experience. I'm sure that this webinar will be a start to more collaboration opportunities, such as maybe conferences, seminars, student activities, and even teaching opportunities. There's more and more to know in this world. And you can only have so much in your head. So people have become more specialized. So this increasing specialization of skills means that you need to have bigger groups with more and more specialists in order to be successful, more successful. So hence to me, collaboration is a must in order for us to attain higher and better achievements. Last but not least, I'm sure everyone will enjoy this webinar with our prestigious speakers and interesting topics, some good knowledge to be shared with all of us today. So with that, I think I'll end my speech with a big thank you to everybody. Hope we all enjoy today's webinar. All right, thank you.
Okay, thank you, Honorable Dean, Dr. Sherija. Moving on, now I request Prof. N. Sivaprasad, Pro Vice Chancellor, GITAM deemed to be University, Hyderabad, India, for the opening address. As Prof. N. Sivaprasad's presence is highly required in today's unexpected meeting at GITAM University, he is sharing the opening remarks through the video.
Can we?
Namaskar. A hearty welcome to Dr. Sharija, the Dean, Faculty of Pharmacy, University Technology, Mara, my colleague, Professor Shivkumar, Principal, School of Pharmacy, GITAM University, and the participants of this webinar. All of you, a warm welcome for this event. Today's topic is challenges and effective drug delivery projects for management of chronic diseases. Is a very important topic. The chronic diseases like asthma, those all seasonally, and how to manage these cases. Sometimes the asthma may lead to even death. And how to manage, how to find out the new drug. Carefully taken the topics like the true, oral, and inhalational and skin drug delivery. That is one. The other one is formulations and approaches to improve the brain uptake by intranasal delivery. For both, intranasal deliveries are there as well as through inhalations are there. Inhalation is basically intranasal, and not necessary inhalation can be to throat also. It will go to the lungs because we keep in the mouth and inhale. That is another way of looking at it. And these are very important aspects in the chronic diseases like the asthma. And in addition to it, is not that the chronic diseases only is the asthma. There are number of chronic diseases. The pharmacy people will be able to identify what are the similar cases and how to treat them and where to use which method. So today, the medicines are becoming part of our food, either through vitamins or through the tablets for the sugar, tablets for the BP, etc., etc. They became as the part of the food because the body system is requiring to balance the diet that we have taken, and the imbalances in our system, metabolic system, are countered by these drugs. And looking at that, the world is facing the pandemic Corona-19 and COVID-19. With that, the whole world, economically as well as psychologically, physically, we are not able to do our jobs. And the scientists, particularly the pharmacists. When we say that the scientists that belong to this medicines are the pharmacists. So after the doctors will use the medicines, whereas the pharmacists will make the medicines. Real challenge to find out the medicine for the pandemic, and I'm happy number of companies, at least 4 or 5 in America, China, Russia, India. These are all the countries which are contributing towards the research, and I'm sure that more than one country will come out with a suitable vaccine for eradicating this. It is in this way, the pharmacy, I'm not sure how big the pharmacy industry is in Malaysia. In India, the pharmacy industry is very big, and Hyderabad is the hub of pharmaceutical industry in India. It is heartening to note that the two universities, that is the GITAM University and the University Technology Mara, have MOU to their pharmacy colleges or the schools to have a collaborative research and collaborative programs for the benefit of both the countries. And research of, exchange of researchers, exchange of faculty members, exchange of students will help a lot in understanding the ongoings in both the institutes and strengthen our bondage between the two universities and also between the two countries. So I wish this webinar a great success, and also the MOU which we have arrived between the two schools will go in a strong way the years to come, and will be able to jointly work and develop the delivery systems, develop the new drugs. I wish all the best to this webinar. Thank you.
Thank you, Honorable Prof. Vice Chancellor, Prof. N. Shiva Prasad. Now I request Dr. Noor Hayati, Head of Pharmaceutics Department, Faculty of Pharmacy, UiTM, Malaysia, to introduce the faculty members of Pharmaceutics. Thank you, Dr. Noor Hayati. I now request Prof. G. Shivakumar, Principal, GITAM School of Pharmacy, GITAM deemed to be University, Hyderabad, India, for introducing GITAM School of Pharmacy, India.
బాక్స్ తీసుకుంటాను.
హైదరాబాద్ మీటింగ్ జూనియర్ ఇక్కడ చేసి పార్టీ దెబ్బకి పారిపోయాడు.
Okay. Yeah, thank you. A warm and a hearty welcome to the participants of this international webinar combinedly organized by UiTM and GITAM School of Pharmacy, GITAM deemed to be university. So in this, I would like to take a brief time to introduce GITAM University and the GITAM School of Pharmacy and the faculty of both the institutions of GITAM deemed to be university. Next slide, please. GITAM deemed to be university, specifically in terms of pharmacy, has a specific vision, mission, and the quality policies. If you could look into the vision, in the coming years, GITAM School of Pharmacy or GITAM and the Institute of Pharmacy is marching ahead towards the visionary ideas which we are focusing towards to become a global leader. And to achieve this global leadership, we are marching towards following the mission of following the infrastructural details and to reaching the mission. So we are moving ahead in terms of following the quality policies, marching ahead towards the vision, mission, and we are strictly following through the quality policies to reach our vision in the next few years. Next slide, please.
Oh.
GITAM recognizes the relevance of Mahatma to all disciplines, ranging from medicine and law on the one hand, to philosophy, architecture, and other humanitarian disciplines. GITAM University ఉన్న కాలేజెస్ ఎన్నో నాకు ఉన్నవాళ్ళు ఉన్నాయి, ఎన్నో ఉన్నాయి. వాటన్నిటిలో కొద్ది కాలంలో #1 ప్రైవేట్ యూనివర్సిటీగా వెలిగింది అంటే
I was coming into the university and I was really impressed with the infrastructure. Fabulous infrastructure.
All the 3 campuses are well equipped and the best in their academic pursuit.
Yeah. Thank you so much for that nice video presentation of the GITAM. Now I would like to introduce the pharmacy department at both the campuses. This campus, Visakhapatnam, we have Visakhapatnam campus and we also have Hyderabad campus. Visakhapatnam campus is led by a very senior professor, Professor S. Ganapati Garu. He is having a very rich experience in terms of 35 years in leading the pharmacy department at both the campuses, followed by the Hyderabad School of Pharmacy. So we have a rich experience followed from the Professor S. Ganapati sir. Next slide, please. Okay, these are the silent features of pharmacy department. We believe in giving quality education in terms of knowledge, practical skills, exposure, training, and also the placements. So these are some of the silent features of pharmacy at both the campuses. Next slide, please. These are all the various accreditations which the GITAM University has been following up. We are being guided through UGC, NAC accreditation, UGC, that is University Grants Commission. The pharmacy is regulated by Pharmacy Council of India, All India Council of Technical Education. We are being ranked at the NRIF ranking, National Institution Ranking Factor, below 75, and we are being rated at the various accreditation bodies. Next slide, please. These are all some of the various bodies where basically the university has been collaborated with. Next slide, please. Okay, so these are basically the research partnerships, the various funding agencies of the India, like Department of Science and Technology, Central Institute of the Industrial Scientific Research, Department of Biotechnology, and so on. These are all the research collaborations that GITAM is having. Next slide, please. Okay, these are all the standard affiliations and the requirements of the innovations which are being provided by the government, like TechWhip and innovation grants. These are all the various requirements that the GITAM University is following up. Next slide, please. Okay, following up with that of some of the industrial collaborations, we have majorly in terms of pharmacy, Dr. Reddy Laboratories, MOUs with that of Divis, Pfizer, Arvindo, Novartis. These are all the companies where our students are getting good opportunities in terms of their job perspectives. Next slide, please. Okay, coming up to introducing to the various faculties. This is a team of pharmaceutics specialization, followed with 2 professors at both the campuses, and followed by associate professor and also assistant professors who are been delivering quality education and pursuing their research and taking up the university and the pharmacy schools at both the institutions at a very higher level. Next slide, please. These are the various research areas of interest of the pharmaceutics department. Next slide, please. This coming up to the pharmaceutical chemistry and pharmaceutical analysis are also followed by both the campuses with the various cadres of faculty. Very high, rich experience of the faculties are being delivering the quality education and pursuing the research for undergraduate, postgraduate, and also the PhD education. Okay, next slide, please. These are all some of the research areas of pharmaceutical chemistry and pharmaceutical analysis where our faculty is carrying out research. Next slide, please. Coming up to pharmacology department is led by Professor S. Ganapati, who is having a very rich experience of 35 years and followed by the faculty in this particular department. Next slide, please. These are the various research areas of interest in the pharmacology specialization. Next slide. Coming up to pharmacognosy, which is natural products and phytochemistry. These are the following faculty who are being serving at both the campuses. Next slide, please. These are the various research interests that the faculty has been pursuing. Next slide, please. Thank you for patient listening. Thank you.
Thank you, Prof. J. Shivakumar, for introducing the GITAM School of Pharmacy to all our participants. After the brief introduction to both the centers of excellence in the field of pharmaceutical sciences in Malaysia and India, we now move on to our scientific sessions. The first speaker for today's webinar is Prof. Wang Tin-Wai from Faculty of Pharmacy, University Technology Mara, UiTM, Malaysia. He will be delivering a talk on the topic Innovative Oral, Inhalational and Skin Drug Delivery for Cancer and Diabetes Treatment. Before the talk, I would like to present a brief introduction of Prof. Wang Tin-Wai. Prof. Wang is currently working as a professor in the Department of Pharmaceutics, Faculty of Pharmacy, UiTM, Malaysia. He is also serving as a visiting professor for National University of Singapore and Yangzhou University, China. He has obtained his first degree in pharmacy and PhD with the specialization of pharmaceutical technology from National University of Singapore. He has a total of 21 years academic experience and specializes in teaching fundamentals and advanced pharmaceutics and drug delivery systems. His research lies in innovative dosage form design and drug delivery sciences, focusing on oral, pulmonary, and skin medicine for the treatment of cancer and metabolic diseases. Till today, 19 master and 9 PhD candidates have been graduated and currently 11 PhD and 9 master candidates pursuing their postgraduate studies in his non-destructive biomedical and pharmaceutical research center. Prof. Wang is a founder and co-director of Sino Malaysia Molecular Oncology and Traditional Chinese Medicine Delivery Joint Research Center, Yangzhou University, China. He has published more than 100 publications in high-impact peer-reviewed journals. He is serving as a regional editor for Current Drug Delivery Journal and associate editor for Drug Design, Development and Therapy and Drug Development and Industrial Pharmacy journals. The latest flash news about Prof. Wang is that he is listed in the world's top 2% scientists in the pharmacy and pharmacology. With this brief introduction to Prof. Wang Tunwai, I am requesting Prof. Wang to start his presentation.
Okay, thank you, Dr. Ravi. Just double check if you can see the slide and hear the voice clearly.
Voice is clear now?
The voice is clear. Can you see the slide?
Yes,
Yeah,
yes.
slide's okay, yeah.
Slide is okay. Okay, thank you. So first of all, I wish to thank Prof. Shariza from UiTM and Prof. Prasad and Prof. Kumar for the kind opportunity to be here for me to share some of the current research finding of our laboratories. So this afternoon, I'm going to share with you on some of the innovative approaches in oral inhalationals and skin drug delivery for cancer and diabetes treatment. Now, skin drug delivery is one of the primary research area of our center, and we focus a lot on cutaneous melanoma. And you can see in this slide, this is a surface morphology of the cutaneous melanoma. Initially, it initiates in the epidermis. With time, it becomes severe. Sorry, the voice is not so clear. Now, with time, it turns severe and it may actually migrate down to the epidermal junction and sooner or later into the dermis and later on metastasize into the systemic circulation. So various drugs and biologics have been used to treat cutaneous melanoma. Now, for topical local therapies, 5-fluorouracil is recently being found to be able to combat skin melanoma. However, drug deliveries to treat skin melanoma locally are faced with challenges whereby both skin and cellular membrane of the melanoma are the drug's transport barriers. Now, in order to understand how to overcome the skin drug delivery, perhaps let's just take a look at the skin anatomy. Now, principally, the skin is constituted of 3 layers: epidermis, dermis, and subcutaneous tissue. Now, the first layers of the epidermis is known as stratum corneum. Now, it represents the primary drug penetration barriers, whether it's to macromolecular therapeutics or for small molecule drugs. Now, broadly, it's known that the drug transport through the skin is mediated through 3 routes. First, it can be taken place through simple transport across the epidermis or stratum corneum. Alternatively, the drug can transport across the hair follicles into the dermis and the systemic circulations or through the sweat glands. However, knowing that hair follicle and sweat gland only occupy 0.1% of the total skin areas. So in the case of pharmaceutics and pharmaceutical technologies, we commonly assume that most of the drugs will transport across the epidermis and stratum corneum. Hence, overcoming hurdles introduced by the stratum corneum is the primary steps to take if we want to succeed skin drug delivery. Now, knowing that stratum corneum is such a barrier to skin drug delivery, perhaps you should understand about its anatomical construct. Now, you can see this in this slide, the schematic diagram of the stratum corneum. Now, basically, it consists of corneocyte, which is keratin-rich, embedded in this lipid matrix. If you look closer onto the lipid matrix, you will realize that it's actually made from lipid bilayers alternating with aqueous pores. Now, further look on the lipid bilayer, you will find various lipid compositions such as cholesterol, fatty acid, and ceramide. Now, I wish to highlight to you, despite lipid bilayers is hydrophobic, now it's named as the lipidic compositions, it does contain hydrophilic and polar moiety, such as OH, NH, and carbonyl group associated with the ceramide, right? So transportation of drugs across the skin primarily mediated through two routes, transcellular and intercellular. Now, a drug molecule needs to penetrate layers after layers of corneocyte if it is to be transported through the transcellular pathway. Now, in the case of intercellular routes, you can imagine that the drugs needs to go through a twist and turn, a very tortuous pathway prior to them reaching the epidermis as well as the dermis. So both transcellular and intercellular drug transportations mechanisms are rather challenging. So it will not be easy to transport the drugs across the skin. So to enable us to successfully deliver drugs, particularly macromolecular drugs, one best way is transiently disturb the systematic structures of the skin, right? So over the past 30 years in pharmacy, we have developed various technologies to overcome skin drug delivery's hurdles. Now, they are broadly classified as passive and active. So the passive technology involves hydration of the skin, whereby applying water to hydrate the skin. The water will enter into the aqueous pore in between the corneocytes. Hence, this will enlarge the aqueous pore size, allowing more drugs to go through. And a lot of skin medicines or even cosmetic and cosmeceutical products, they do contain chemical penetration enhancer, such as oleic acid, ethanol, and surfactant. Now, this penetration enhancer is able to fluidize lipid at the intercellular routes, or they may even extract and get rid of the lipids in order to form a larger aqueous pore for better drug penetration. Now, nanocarrier, of course, is the latest favorite of all scientists. Now, you will note that the nanocarrier, indeed, it can be made from waters, it can be made from oleic acid, ethanol, and surfactants. So with these chemicals, the aqueous pore can turn larger, allow more drugs or even more nanocarriers to go through. Now, of course, nanocarrier has a very unique features in the sense that it is small, it's nano in geometry. Now, it itself can actually squeeze through the intercellular routes and penetrating into the dermis and systemic circulation. Now, what about active technology? Of course, conventionally, we have the 3M adhesive tape to remove the stratum corneum. This is an outdated technique. Microneedle is one of the current hot topics, whereby skin is porosified by needles to deliver the drugs. Electrical methods such as iontophoresis, electroporations, ultrasound, sonophoresis, photomechanical wave, and pressurized gas are some of the approaches that have been investigated by various scientists globally. Now, let's take a look on how nanocarrier deliver drugs across the skin. Now, you will see that upon applicating nanocarriers or nanomedicines onto the skin, now the nanocarrier may be released the drug on the surfaces of skin, and the drugs will then diffuse downwards into the epidermis and dermis. Or alternatively, the nanocarrier may be fused with the upper membranes of the stratum corneum, and the drug is released at the upper surface towards the lower surfaces of the skin. Last but not least, of course, the nanocarrier being small and it has a sufficient elasticity, it will be able to squeeze through the intercellular routes and penetrates into the through the intercellular routes into the epidermis and dermis and release drugs at the lower parts of the skin. Now, skin drug delivery by nanocarriers is rather challenging. Now, primarily because the size and the zeta potentials of the nanocarrier will dictate how much drugs can actually go through the skin. Now, in our laboratory, we have conducted a systematic review over, I think, about 200 articles. Now, we realized that the relationship between skin drug transport as well as size of the nanocarrier is not straightforward. Now, we actually examine vesicular carriers such as liposome, attosome, transposome, polymersome, nanoemulsion, and polysaccharide nanoparticles such as alginate nanoparticle, chitosan, and pectin nanoparticles. Now, you realize that unlike what we believe, that smaller size will allow more nano as well as more drugs to go through the skin, but the review outcome shows otherwise. You will see that a lot of small nanoparticles, they actually have a very low skin drug transport propensity, right? And the slightly larger nanoparticle, indeed, they may have a larger skin drug transport propensity. So the relationship between skin drug transport and nanocarrier size is not simple and straightforward. Now, the same applies to zeta potential, surface charges of the nanocarrier. Now, with reference to vesicular carriers such as liposome, attosome, transposome, there's no direct relationship between the skin drug transport versus the zeta potentials of the carrier. Now, in the case of nanoemulsion, we notice that minus 10 to 20 millivolt nanoemulsion is able to transport the drugs better across the skin. Whereas for polysaccharide nanoparticle, we have interesting findings. You will see that positively charged as well as negatively charged nanoparticles are able to convey the drugs or the nanocarriers with a higher propensities to go across the skin. Now, this will not be difficult to imagine. Now, for positively charged nanoparticles, they will be able to attract to the skin layers as the skin is constituted of anionic lipids. So opposite charge attract. So on that note, the nanoparticles, the drugs will be able to deliver across the skin easily. Now, what about negatively charged nanoparticles or nanocarrier? Now, being negatively charged, they are able to repel the anionic lipids of the skin. Now, consequently, the skins will turn deformable, softer. If you can imagine the intercellular routes, the lipids domains may be distorted, thus forming a larger aqueous pore. Now, on this basis, hence the drugs that are released earlier on the surface of the skins may be able to penetrate through the skins easily. Or even some of the nanocarrier or nanoparticles may be able to penetrate through the skin because of the deformed lipid matrix at the intercellular route. Now, in year 2000, our laboratories begins to use microwave, not for skin drug delivery, but for processing of solid medicine, such as microparticles, polymeric bits, tablets, pellets, and granules. Now, what we have found about the microwave is it's able to interact with polysaccharide, protein, and lipid materials of the solid medicine. Now, we note that the microwave can actually enhance the conformational flexibility of polymeric substances. Now, with reference to the skin composition, you note that in the earlier slide, they are made from lipids, a lot of lipids, and also keratins and proteins in the corneal side. So we will wonder, perhaps the microwave can do the same to the skin. Perhaps it can fluidize the lipid zone and hence making larger aqueous pore to allow more drugs and nanocarriers to go through the skin, right? So this is our initial hypothesis. Now further, through our readings, we also realized that microwave has two components, thermal and non-thermal. So in the subsequent investigative study, we wish to capitalize on the non-thermal components and ability of microwave to fluidize, to make the polymeric substances more flexible as an anchor to enlarge the aqueous pore, right? For a better skin drug delivery. So we have carried out a preliminary study. Now in this study, what we have done is we harvest the rat skin and we treat the skin with microwave from the directions of the epidermis. Now you can see that treating the surfaces of the skin with the right frequencies microwave, 2450 megahertz. Now instead of 3985 megahertz, and the right duration, 5 minutes instead of 2.5 minutes, the surfaces of the skin turn smooth, right? Compared to the untreated skin. So this actually alarm us perhaps the microwave really fluidize certain materials in the skin, and perhaps the systematic structure of the stratum corneum is being destroyed temporarily to allow more drugs to go through the skin. So with all these readings and preliminary studies in mind, we move on to create anti-cancer drugs deliveries through the skin. Now, in these studies, we adopt 5-fluorouracils as the drug of interest. This is because the 5-fluorouracil has earlier been reported that they are able to treat the skin melanoma rather effectively at the clinical trials levels. However, note that 5-fluorouracil, though it is small in size, it can actually encounter penetrations hurdles due to its hydrophilic natures, right? So in this study, we formulate the 5-fluorouracils into atosome. And atosome is selected because atosome has been found to be able to improve skin drug retentions relatively better than other nanocarriers such as liposome or even solid nanoparticles, right? So these studies, principally, we wish to deliver 5-FU in the form of atosomes to treat local skin melanoma. So it's meant for topical treatments, not for systemic treatment. So 3 types of atosomes have been formulated. So E5, E10, and E15 represents the atosome with low ethanol content, 5%, and higher ethanol content, 10 and 15%. Now we realize that when the atosomes contain a higher levels of ethanols, it will release the drugs easily, and at the same time, upon application of atosome onto the skin, you realize that a lot of drugs can actually penetrate into the systemic circulations. So this is highly unfavorable because we wish to treat the melanoma topically and locally instead of systemically, right? So E5, among all variants of atosome, is found to be favorable. So you will see that E5 with a lower ethanol content, it does raise the systemic permeations of the drugs, but lesser than that of E10 and E15. But one great advantage of E5 is it actually raised the skin drug retentions. Now in the epidermis or the upper dermis, and this is favorable for local melanoma treatment. Now, an examination of the skin microstructure that are applied with atosomes by FTIR imaging. Now, we realize that the atosomes are able to accumulate in the epidermis when E5 is used compared to E10 and E15. Now, when E10 and E15s are being adopted as the delivery vehicles, perhaps due to the high ethanol content, they are able to penetrate so well through the epidermis and dermis and has been entering into the systemic circulation. Hence, you can't really find it in the epidermis and dermis in a sufficient quantity, unlike E5. Now, E5 is able to be retained in the epidermis, and some of them, you can see the red little dots in the dermis. One possible reason is because E5 is generally larger in size. It has higher levels of negative charges. So with these higher levels of negative charges, this E5 is able to soften the lipid components in the stratum corneum to a greater extent. Now, it may be able to enter into the skin, but it will not be able to go deep down into the deep dermis or the deeper skin layers, and hence disappear into the systemic circulation. Right. And you may wonder that if we want to improve the skin drug retention specifically for topical melanoma treatments, why not we prepare E2.5 or E1 with a lesser content of ethanols? The answer is no, because in the attosome design, we need a minimum level of ethanol in order to keep the attosome elastic, able to squeeze through the intercellular routes. So we keep E5 as the optimal compositions. Now, in order to further retain the drugs as well as attosomes in the epidermis or the upper dermis of the skin, we actually combine the use of attosomes with microwave. Now, we're trying to see what is the outcome if we have a combined treatments on the skin. So you can see that if we treat the skins with microwave at 2450 megahertz for 2.5 minutes, followed by the applications of attosome, there will not be any significant rise in the systemic drug permeation. However, the skin drug retention is greatly enhanced when we treat the skin with microwave with the right frequency and the right durations prior to the applications of E5. So this finding is really exciting. And we run against the FTIR imagings to confirm that the attosomes is indeed being located in the epidermis or also in part of the dermis. Now, to enable us to understand why does the use of microwave to treat the skins will enable more drugs and attosome being retained in the skin instead of penetrating into the systemic circulations, we characterize the skin by a few techniques. First and foremost, we characterize the lipid and protein regimes of the skin by means of thermal analysis. We also characterize the epidermis and dermis of the skin that is treated with microwave, with attosome, or with various chemicals of the attosomes by means of FTIR. Now, to make the interpretation simple, instead of going through the values, allow me to use these schematic diagrams to demonstrate to you how the microwave can influence the skin drop penetrations of an attosome. Now, in the earlier slide, I've highlighted that the stratum corneum is constituted of corneocyte and separated by the intercellular roots that is containing of a lipid matrix as well as the aqueous pore. And of course, stratum corneum is part of the epidermis. It is supported by the dermis. Now, treating the skin by microwave will bring several changes to the skin microstructure. First and foremost, the microwave is found through the thermal and FTIR study is able to actually transform the keratin in the corneocyte from alpha helix to beta sheets. So as a result, the keratins will condense. Now, once the keratins in the corneocyte condense, the larger corneocyte will shrink into the smaller corneocyte. Now, concurrently, the microwave also interact with the lipids in the intercellular roots. So the lipids will be fluidized through mobilizing the hydrocarbons packing. The summative effect is that the intercellular roots of the skins will turn larger, right? After treating with microwave, this will then allow drugs and attosomes to go through to penetrate the skin at a greater ease. Now, incidentally, the microwave can also interact with the dermis of the skin. Now, from our study using FTIR and differential scanning calorimetry, now we tentatively conclude that perhaps the microwave can actually interact with the extracellular proteinaceous matter such as elastin, and we suspect that these proteins matters will swell. Now, having a swollen dermal materials, you can imagine that they will form a barriers below the epidermis. So, in conclusion, the drugs and attosome can penetrate well through the skin due to the enlarged aqueous pore, but they will not be able to go deep down, into the lower layers of skin as a result of a formation of a dermal barrier. So overall, there will be more drugs being retained in the skin, and there will be less drugs or less tendency of drugs appearing in the systemic circulation, right? So this is the very first mechanism that has been found by our laboratories, and more works are going to be done, in order to elucidate the more exact mechanism. Now, in order to further verify our in vitro finding, we actually carry out in vivo experiments and using the rat models. Now, the rats, the skin surfaces of the rat is treated with attosome, and in another group of rats is treated with microwave at the right frequency for the right duration, followed by the application of attosome. Now, you will see that the drugs has a less tendency to appear in the systemic circulations when a combinations technology is used, and a lot of drugs instead are being kept in the skin layers. So these in vivo findings indeed support the in vitro's outcome, and it also indicates that the combination technology of attosome and microwave can be a potential tool for topical treatments of melanoma. Now, what about melanoma? Now, having succeeding to deliver the drugs across the skin, will the drugs be able to enter into the melanoma and exert its cytotoxic action? The answer is yes from this slide. Now, you can see that the drug-loaded attosome, as well as the microwave alone, indeed, they are able to exert a higher level of cytotoxicity compared to the neat drugs, right? And combination use of microwave followed by attosomes bring about synergistic actions against the melanoma growth. And we run the thermal and infrared analysis on the melanoma cell line that is treated with microwave, with attosome, or combinations of attosome and microwave. Now we realize that attosome itself or microwave itself, they can actually fluidize the lipid materials of the membrane. And combination of attosome and microwave will aggressively fluidize the lipid membrane, the lipidic composition of the membrane through interacting with the phosphate moieties available on the melanoma membrane. So this fluidization of the melanoma membrane will allow more attosome and drugs to penetrate into the cellular compartment and exert the cytotoxic action. Now, in the next slide, you will see the confocal electron microscopy data. Now, the cell line that is treated with microwave followed by attosomes appears to have more attosome being migrated into the cellular compartment. And using the same experimental design, we also pre-treated the cell line with various metabolic inhibitors such as cropamazine, nystatin, genistein, and warfarin. Now, we realized that the microwave can actually facilitate the entry of attosome into the melanoma cellular compartment through lipid raft endocytic pathway. So this finding is rather new, and this finding is also corroborate with our skin drug delivery finding, whereby microwave can actually interact with the lipid material of the intercellular roots, right? In order to fluidize them to enable more penetration of drugs across the skin. Now, in another studies, similarly on topical treatments of melanoma, instead of a liquid attosome, we designed solid chitosan 5-fluorouracil conjugate nanoparticles. Now, the same findings is being found. Now, treating the skin with microwave at the right frequency and with the appropriate timings, you can see that more nanoparticle can actually enter into the skin. Hold on. You can't move the slide. All right. Now, the combination use of the nanoparticle, simple chitosan nanoparticle with the microwave bring about synergistic effect to deliver the drugs across the skin for local treatment of melanoma. Now, one of the reasons that we have found out is because the chitosan nanoparticles, possibly due to the chitosan natures, it can actually fluidize the palmitic acid of the lipid domains, whereas the microwave is actually fluidize the ceramide 2 of the skin lipids. So you can see both chitosan nanoparticles and microwave, they fluidize a different lipid composition. So combinations of both efforts will actually synergize their skin drug delivery capacity. Now, in the same study, we further modified the chitosan nanoparticles. Now, in this study, we actually designed chitosan 5-fluorouracil nanoparticles with folate conjugate to it. Now, folate is a targeting ligand for folate receptor that are available on the surfaces of the melanoma membrane. So using chitosan 5-fluorouracil folate conjugate nanoparticle, the same outcome has been derived. You can see that more nanoparticle can be accumulated in the skin layer for treatment of cancer with concurrent use of microwave as a skin penetration enhancer. And the microwave, similarly to the liquid attosome, it actually helps the solid nanoparticles to enter into the cytoplasms of the melanoma through lipid raft endocytic pathway. Right? Now, folate is used in the late study. That's because apart from topical treatments of melanoma, we actually also intend in the future look into the use of chitosan nanoparticle to treat the cancers systemically, right? So folates being targeting ligands to the folate receptor, it play a very important role in order to bring the cytotoxic drugs only to the intended target, that is the melanoma cell. So in one of the recent study, we have found out that 20% folate to conjugate nanoparticles is the ideal composition of folate in order to enhance the intracellular uptake of the nanoparticles into the melanoma cells. Now, with reference to skin drug delivery, now apart from cutaneous melanoma, our recent attempt is to deliver the macromolecular therapeutic insulins through the skin. Now, conventionally, in skin formulations for the purpose of transporting insulin, now oleic acid, linoleic acid, and linolenic acid are used as a chemical permeation enhancer in order to fluidize the skin and enable more drugs or more insulin to go through the skin. Now, in the late studies of ours, we found we have a slightly different findings. Actually, we have found that the fatty acid, be it oleic, linoleic, or linolenic acid, they demote transdermal insulin delivery instead of promote, right? Whereas microwave can actually promote the transdermal insulins to a significantly higher extent than those of fatty acid. Now, this study is really exciting us because this shows that perhaps the combination use of microwave and insulin formulations, you will be able, how should I say, to be transported across the skin now without the need of the fearful injection. Now, what is the reasons why the microwave can improve skin insulin deliveries in comparisons to those of the fatty acid? I think there are two components and two elements that we need to remember. Now, first of all, the microwave can improve the skin permeability. They may be able to shrink the corneal side and to fluidize the lipid layers in the intercellular roots, thus making a larger aqueous pore. This is further illustrated through a transepidermal water loss experiment. So in this experiment, treating the skin with microwave is accompanied by a high level of transepidermal water loss. So the microwave can actually improve the skin permeability. And more importantly, microwave compared to fatty acid, it is a residual-free approach. Unlike fatty acid, which deposits a hydrophobic shield, which is highly unfavorable for skin transport of insulin. Now, to enable us to understand a little bit more about the complication that is brought about by fatty acid, if you look at this diagram, upon treating the stratum corneums with microwave, we have a larger intercellular route favorable for insulin delivery. However, if in any exercise that fatty acids is applied, now this fatty acids is found to be accumulated within the intercellular route, and it forms a hydrophobic shield or hydrophobic barrier. And knowing that insulin is a large molecule drug and it is highly hydrophilic, hence it will be very difficult for the hydrophilic and large molecular weights insulin to penetrate across the intercellular routes due to the hydrophobic natures of the intercellular route, right? Although the intercellular route has been enlarged, but due to the hydrophobicities, the insulin will not be able to penetrate successfully. Now, let's go to the inhalational drug delivery. So we have recently moved into inhalational drug deliveries, and our experiment is rather simple. We synthesize our own nanomaterials, and following that, we design the nanoparticles, principally decorated with targeting ligands. And in order to deliver the nanoparticles successfully into the lung without being exhaled, and we actually also design microcarrier that can carry the nanoparticles into the deep and peripheral lung. And we hope that in the deep and peripheral lung, the nanoparticle will be released from the microcarrier. A nanoparticle can either act directly onto the cell or being transported into the cytoplasm and exert the cytotoxic action. Right. Now, over the past 10 years, due to the exhalation tendency of the nanoparticles, various researchers have designed various microparticles for inhalation. Principally, we have a nanoagglomerate, nanocomposite, and nanoparticle being encapsulated in the microparticles. Now, all these nanoparticles experience poor redispersibility. Hence, we may not get any nanoparticle in the deep and peripheral lung. And a lot of times, for example, due to the polymeric substances encapsulating the nanoparticles, you may find it the nanoparticle or even the drugs cannot be released for the intended action. And the worst still, in the process of making microparticles, your solvent, your polymeric material may actually distort the nanoparticle size of the nanoparticles. So this is highly unfavorable because uptake of nanoparticle by cancer or even by the normal cells is depending on the size features. Right. So on this note, we actually hypothesize a possible way to deliver the nanoparticles in a better capacity. Now, if you read through the articles for the past 20 years, now the small particles usually is deposited on the coarse microparticles. And this coarse particle will then be inhaled in order to deliver the drugs into the deep and peripheral lung. And note that the coarse particles is having a size of 200 to 300 micron in general. So more often than not, the nanoparticle will not be able to detach from the coarse microparticle. And coarse microparticles being few hundreds micron in size, it will not be able to reach the deep and peripheral lung. Usually is situated at the back of the throat. So when this occurs, there will not be any possibility for the nanoparticle to be inhaled into deep and peripheral lung. As such, we actually hypothesize that we should actually deposit our nanoparticles on a micro scale particle, which is a few micron size. Now let's take the hypothesis that we have derived. Now you can see that nanoparticle alone, due to the small size and high specific surface area, they tends to be aggregative. Likewise for micro scale particle, because it is only a few micron size, they are cohesive and aggregative. So blending of both in a specific weight ratio, you will have the nanoparticles being absorbed on the surfaces of the micro scale particles. Now note that once the nanoparticle is absorbed onto the surfaces of the micro scale particles, the aggregativeness of the nanoparticle is reduced. And the nanoparticles sitting on the surfaces of the micro scale particles, it also can act as a glycan, thus preventing the microparticles aggregating with one another. Right? So this system appears to be ideal for simple reasons. Upon aerosolization inhalations, the particles can actually reach the deep and peripheral lung due to the small size in nature. And in the deep and peripheral lung, the nanoparticle can detach from the microscale particles and act on the cancer cells, for example. And even though the nanoparticle is not detaching, but at least the whole assembly of the particle will reach the deep and peripheral lung, right? So on this note, we deem that microscale particle is definitely better than the coarse particle. With this hypothesis in mind, we actually prepare 6 variants of solid chitosan nanoparticle by spray drying. We vary the acetic acid concentration, chitosan grade, molecular weight, chitosan concentration. Also, we use different excipient, twin, span, and ethanol in order to prepare 6 different types of chitosan nanoparticles with different physical-chemical properties. Now, the physical-chemical properties of nanoparticles that are being evaluated are size, zeta potential, polydispersity index, circularity, the shape, the surface roughness, as well as the matrix crystallinity. Now, these properties are evaluated because they are the features that will dictate the nanoparticle inhalation's efficiency. Now, you can see that the nanoparticles prepared are rather round in shape. And we also prepare our own microcarriers. Now, instead of using the commercial lactose inhalations grade, we actually prepare our lactose microcarrier through incorporating polyethylene glycol 3000. Now, this PEG 3000 is very important because it is waxy in nature. It can reduce the hygroscopic nature of the lactose particles. So the lactose PEG 3000 microparticles is only several micron in size. Now you can see the shape is definitely not round, it's a rock shape. And through XRD analysis, you realize that our spray dry lactose PEG 3000 particle is rather crystalline compared to those of the pure spray dry lactose. So this will be good because it reduces the hygroscopicity, which may complicate our inhalation efficiency. Now, blending of the microscale particles as well as the chitosan nanoparticle in a specific rate ratio, we then move forwards to conduct a cascade impaction study to simulate the aerosolization and inhalations process of the nanoparticles. And from this exercise, we realized that the size, zeta potential, as well as the circularity of the nanoparticles dictate the inhalation efficiency of the drugs. Now you can see that when the nanoparticles is larger, is rounder, they will have a lower specific surface area. So the nanoparticles will not interact among themselves, but instead the nanoparticles will be absorbed and sit on the surfaces of the microscale particle. Thus, this will enhance their inhalations into the deep and peripheral lung through the help of the microscale particles. And nanoparticle that has a higher zeta potentials, they tend to repel between each others. So there will be a lower level of nanoparticle aggregation. Most of the nanoparticle will absorb on the surfaces of the microscale particles and being brought into the deep and peripheral lung. Now, on the contrary, if the nanoparticle is very small, not circular in shape, they will exhibit a large specific surface area. Now, this nanoparticle tends to aggregate between them instead of absorb onto the microscale particles for transportation into the deep and peripheral lung. So likewise, if the nanoparticle has a lower surface charges, lower zeta potential, they will aggregate between them instead of interacting with the microscale particles for inhalation, right? Now, so far, for the solid chitosan nanoparticle, we also actually design liquid nanoemulsion for inhalational studies. In this study, we actually prepare first, second, and third generations nanoemulsion. The first generation is very simple. Basically, oleic acid, lipid components for us to prepare oil in water nanoemulsion. The second generation, we have a chitosan decorated on the surfaces of the oil droplets. And the third generation, we have chitosan folate, the conjugate, decorated on the surfaces of the oil droplets. Now, chitosan or folate are being used because they are targeting ligands for the macrophage. Now, chitosan can actually recognize the mannose receptor on the surfaces of the macrophage, whereas folate can actually recognize the folate receptor on the surfaces of the macrophage as well as the cancer cells, right? Now you realize that once we decorate the oil droplets with chitosan folate conjugate, you will see that in the macrophage, a higher level of nanoparticles are being engulfed into the macrophage. Now, as a result, what we can see is that the lung drug content will be higher when we deliver drugs by means of third generation nanoemulsion. So the use of chitosan folate conjugates are able to allow more nanoparticle interacting with the macrophage, and hence more drugs being uptaken by the macrophages, right? So such conjugates, due to the availability of two targeting ligands, we name it as dual receptor targeting. Now, let's move on to the final section on oral drug delivery. Now, in our laboratory, we have been doing drug delivery right from the year 2003. So it has been about 17 to 18 years. Treatment of colorectal cancer by means of oral colon-specific delivery is our main interest in the laboratories. Now, in these studies, we actually create in vivo intracapsular coating instead of fluid-based or immersion coating. So we have our cancer therapeutic pellets being coated in the body instead of being coated using the machine on the bench top. So let me go through with you on the design briefly. Now, first of all, we actually load the drug into pectin-rich pellets, and we mix the pellets with coating powder, which is constituted of hydrophilic pectin, as well as the hydrophobic ethyl cellulose. So mixing of pellets and coating powders is run in the hard gelatin capsule. Now, upon consuming the hard gelatin capsules into the biological fluid through the gastrointestinal tract, now you realize that in the gastric medium, the coating pectins, due to its hydrophilicity, it will be wetted by the water, so it will turn adhesive, and it will bind the ethyl cellulose to the pellets and form a plug. And this plug is rather resistant to disaggregations along the intestinal transit. Now, however, towards the terminal ileum, the terminal intestinal regions, the plug will begin to disaggregate with time. And in the colon, what happens is that we have a lot of microbial enzymes, and this microbial enzyme can actually digest the pectin of the pellets, as well as pectins from the coating powder mixture. So digestions of the pectins results in disintegrations of the pellets, and the total release of the drugs will occur and takes place in the colonic region. So this study is rather interesting because it adopts a very simple technology, and hence you can actually achieve the same outcome. Now, what interests us more is about the pharmacodynamic performances of intracapsular in vivo coated pellets. Now, these pellets carrying a 5-fluorouracil is able to reduce the size, the number of the colorectal cancer in vivo. It's also able to bring the histopathological grades of the colorectal cancer back to normal. But what interests most is that 5-FU, 5-fluorouracil, that is released from intracapsular coated pellets, in addition to combat the cancer growth, it can actually also reduce the aberrant crypt foci that is found in the colonic region. So this aberrant crypt foci is the preneoplastic lesion. Now, it's the cell population that is about to turn cancerous. So you can see the intracapsular coated pellets is not only just able to kill cancer, it can also kill the precancerous lesion. And this will be very useful for us to totally eradicate the colorectal cancer. Now, in other studies, instead of engaging coating mechanisms, we actually try to formulate pellets that can deliver drugs to the colon without being coated. So we presume that without coating, this will actually save the materials as well as the energies and time of the manufacturers. Now, to cut the story short, Now, in this study, we actually prepared the matrix using alginate. Now, we loaded with ethyl cellulose and calcium phosphate. Now, the ethyl cellulose is very important in the initial part of the gastrointestinal transit because due to its hydrophobic nature, it can actually prevent the water ingress into the pellet structure, and hence less drugs will be released in the upper gastrointestinal tract. Now, what about calcium phosphate? Now, calcium phosphate is water soluble, but it is a moderately water soluble calcium salt. Now, it will be solubilized only gradually, and this gradual solubilization of calcium phosphate is very important because it will sustain the alginate matrix through formation of calcium alginate structure. And this structure will be rather robust across the gastric and towards and across the intestinal region. And it is only when the whole matrix entering the colon, the structure will be broken down into the smaller particles for site-specific drug release. Now, calcium phosphate is very important. If we use the wrong calcium salt, such as calcium acetate, early disintegrations of the matrix will occur. This is because calcium acetate is highly water soluble. No doubt, high water solubility of calcium acetate will promote calcium alginate formation, will allow a more robust structure to form. But the fact that fast solubilization will create dispersive forces. So this turbulence and dispersive forces will break the alginate matrix into smaller particles that will render premature drug release. Right? Now, in addition to transdermal insulin, we actually also have an oral insulin project. Now, again, alginate is being used. Now, in this project, we use alginate for multiple purpose. Now, alginates, it has been a long time, known by the ethnopharmacologists that it's a sugar absorber. It can interact with the sugar in the gastrointestinal tract and helps to keep the sugar in the intestinal lumen instead of being absorbed into the bloodstream. So alginate, in a way that it does possess some anti-diabetic properties in its own way, right? Now, in the experiment, we also engage alginates as the encapsulating materials for the oral insulin. So we prepare alginate carbon 18 conjugate to encapsulate our oral insulin. Now, alginate alone is not used because alginate can interact with the insulin in an excessive manner to the extent that the insulins cannot be released and it cannot even be detected by the instrument. Through designing alginate in the form of alginate carbon 18 conjugate, this conjugate is able to encapsulate the insulin, protect them, but yet release them when needs to. So alginate carbon 18 conjugate nanoparticles is later being loaded into the calcium alginate bits, which is larger in size, about 1 to 2 millimeter. And you note that the bits materials is the alginate. That's because bits material is available in a larger quantum. So using a larger amount of alginates, it can also confer sugar absorbing property that will help us to reduce the rise in blood glucose in the systemic circulation. And the entire nanoparticle in bits is coated with chitosan oleic acid conjugate in order to protect them from early release in the gastric phase. Now you can see that oral consumption in vivo of these structures enable good glucose lowering due to enhanced bioavailability of the insulin in the systemic circulation, right? And looking at the in vitro drug release data, however, we found something unexpected. Now you can see that our coated bits structures is able to reduce the early release in the gastric regions. However, in the intestinal regions, only about 30% of the insulin is being released. But nonetheless, a high bioavailability of insulin is observed in the in vivo study. So the main reason for it is the insulin is being delivered into the systemic circulation, not just by free insulin absorption transmucosally. Now the insulin is also absorbed through penetrating through the intestinal mucosa and entering the bloodstream thereafter. Now you can see in this slide, the nanoparticle that we prepare has a very low level of cytotoxicity. It can actually transport across the mucous batters and being trapped by the intestinal mucosa prior entering into the bloodstream to exert the anti-diabetic action. So the uptake of nanoparticles of ours by intestinal mucosa is mediated by cavular and claritrin endocytic pathway. And this will be my last slide, but it's our first very successful attempt to formulate the herbal active into the form of nanoparticles for intestinal targeting in diabetes treatment. So we have been research on Ficus deltoideus for the purpose of anti-diabetic action. Now, through these few years of research, we have identified vitaxins being vital components to lower the blood glucose via its alpha-glucosidase inhibitory action. Now, on this note, we then tries to develop nanoparticles as the carrier for vitaxins and try to deliver vitaxins to the intestinal region specifically in order to interact with the intestinal alpha-glucosidase. Now, how we do it? We have the alginate and stearic acid mixture, and we mix it with vitaxin. We spray dries into the nanoparticles, and subsequently we actually compact the nanoparticles with polyethylene glycol 10,000 into the form of the compact, right? And this compact is being inserted into, it's a very small compact and being inserted into the capsule and administered in vivo for oral anti-diabetic action. Now, what we have found out is that the polyethylene glycol function very well to reduce the vitaxin release to almost 0% in the gastric region. Now, upon compact entering into the intestinal region, you can see a surge in drug release. So the small molecule vitaxin is very hard to control, right? But in our case, we are able to reduce the release in the gastric phase to 0% and enhance the release in the intestinal region. Now, in the intestinal regions, what happened exactly is that the PEG will be eroded with time and in the intestinal fluid. And very importantly, the nanoparticles is constituted of alginate and stearic acid. Now, the alginate is the polyanionic polymers. So in the intestinal fluid, it will experience ionization, losing its proton. It will carry a negative charge and the alginate will repel between each other and the nanoparticles will be dispersed or fragmented. And stearic acid also has its role whereby it will induce wicking. It can actually hit on the nanoparticle matrix and try to disaggregate them in the intestinal region. Hence, more vitaxin will be released in the intestinal region per se for vitaxin targeting on the alpha-glucosidase. Now, you can see that if we replace alginate stearic acid with alginate alone, there will be no favorable pharmacodynamic performance. Likewise, now if we replace alginate stearic acid mixture with alginate stearic acid conjugate, the ideal pharmacodynamic actions will not be attained, right? Alginate alone, it will not function very well because there's no wicking action of the stearic acid. And conjugating the stearic acid onto the alginate, you actually restrict the stearic acid movement. So the wicking action of stearic acid is not there. And hence your nanoparticles cannot be dispersed so well in the intestinal regions to release the vitaxin, right? So much so for today. And again, I wish to thank Prof. Shariza, Prof. Prasad, Prof. Kumar from GITAM, my postgraduates, and one of them have become, I think 4 of them have become the lecturers. Dr. Nokaizai Anwar is with us, Dr. Nauman, Dr. Asif, and one Dr. Muham. They are now working as a lecturer in Pakistan and Malaysia. So with that, I thanks again for the organizer for giving me the opportunities to be here and hope to see you in the future. Thank you.
Thank you very much, Prof. Wang.
Okay.
The presentation is very interesting and incredibly informative.
Thank you.
Now we are opening the question and answer session. Dear participants, you can unmute the audio and ask the question directly to Prof. Wang, or you can also post the questions in the chat box so I can ask behalf of you. Thank you.
Is that too fast just now, or is the sound stable? Because I can hear the disturbances through the headphone.
Yeah, sound is stable, Prof. Sound
Okay.
is stable. Yeah,
Thank
it's
you. Thank you.
it's very clear.
Thank you.
Any questions to Prof. Wang? You can also post in the chat box so I can ask behalf of you.
I think Prof. Wang has given a good talk. That's why
I think most of them should be undergraduates, right? Dr. Sanita? Yeah, sure.
Thank you for your presentation. Very, very informative. I just have a quick question. Regarding your transdermal delivery of 5-fluorouracil for melanoma, you mentioned that one of the purpose of your design was to prevent a systemic delivery rather to induce or localized delivery of the drug towards the melanoma cells. So I was just wondering, in your personal opinion, because most melanomas are identified or characterized early on, that means they are identified at early stages, right? So that means if they haven't broken the, so to say, the basal membrane, that means they have not metastasized to any other locations. So most of them are excised, and usually the recovery rate is quite satisfactory. So in that sense, in your personal opinion, how does that your particular design of drug would improve our treatment of melanoma in the future?
I think for us, we only target on melanoma at the early stage, right? So I think if you look at the studies, I've been thinking, would it be wise, would it be profitable if we aims to use the attosome and microwave combination technology for late melanoma stage where metastasis has occurred? The answer, most likely not. I'm thinking perhaps microwave in combinations with nanocarrier should be used for the early stage. You can see that the microwave itself also has cytotoxic properties. So application of microwave, I would say, should be directly onto the melanoma, which is visible on the surfaces of the skin. So this will enable us to do the first eradications of the cancer, now followed by the applications of nanocarrier, such as attosome or solid nanoparticles. So pertaining to your question, I would say that I think early stage is good. Late stage, I think is rather unfavorable, and perhaps we can combine with oral medications instead. Yeah.
Okay, thank you.
Very good afternoon, Professor Wang. Thanks, sir.
Hi, afternoon.
I have a so when you are using this particular microwave assisted complex, how about the stability of the product?
The voice is breaking, right? I'm not sure. Is that only me?
Okay, can you hear me right now?
Okay, I can hear you, but you may need to speak at a lower volume because your echo is very high.
Okay.
Yeah, yeah.
Is it okay?
Yeah, it's clearer now. Thank you.
Yeah. Yeah. The question I have is when we are preparing the ethosomes, yeah, using this microwave assisted formulation approach. So can you just tell me the stability aspects of the ethosomes?
Okay. Now, if you look at the entire study, be it transdermal, inhalational, or oral drug delivery, you note that I have never indicated any information about the stability. That's because the centers that we have in Malaysia, in UiTM, we actually tries to focus more on innovative ideas. So our approach is very simple. First, to prove the innovation or innovative ideas, the stability should come later. I do notice that a lot of researchers right away they will do the stability test and things like that. But in my opinion, this is not advisable. I would say that until an idea is proven, then we then will spend time looking at the formulations, optimizations to enable a highly stable products being produced. Yeah. So this is just my personal opinion. For example, I also realized that a lot of papers from India, a lot of them, they have a good ideas and right away they move into stability tests. And personally, I feel that it's not really necessary. Perhaps the researcher can spend more time on the mechanistic aspect of the formulation performance. This will be much needed by the current journals.
Thank you.
Thank you.
Any other questions? Profon. Although I am a moderator, I am also having one question.
Okay, okay. That's a common strategy. If no one question, moderator has to ask. Yeah. That's why I do not like to be the chair or the moderator because you need to pay attention.
Yes, of course. Agree with you. What exactly the mechanism involved in disintegration of intracapsular coating in the colonic region compared to the immersion coating? How it is better over the immersion coating?
Emulsion coating.
Yeah, immersion coating, fluidized bed and immersion coating. Yeah.
Okay. So I can share with you some of the past experiences by the UK and European researcher. Now they prepare 5-fluorouracil pellets and they coat with, I think the famous chemical is eudragic.
Yeah. Okay.
And some of them also mix the eudragic with pectin as the coating materials to the pellets. Now, pectin is needed in the eudragic coat so that when the whole pellet system reach the colon, the microbial enzyme can digest the pectin in the coat. Then you can form a pore. And this pore will allow the drugs to leach out into the colon. Now, however, the fluid-based coating of eudragic pectin coating materials is met with difficulty. Now, what are the difficulty? First and foremost, the pellet successfully reached the colon. The pectin in the coat leach out. The pores are formed just before the drugs can release. Due to the glass transition temperatures influence and others solvent polymer interaction, the eudragic reform and the eudragic readjust themselves to seal the pore. So principally, there's some difficulty in releasing the drugs in the colon. Now, if you look at our design, we do not coat by fluid bed, but we adopt a powder system, pectin and ethyl cellulose, and they actually hold them in a loose form, but a very robust aggregate, or you can call it as a plug, right? And using this system, when the whole system reach the colon, they can disintegrate relatively easily without being affected by the physical chemical property of the ethyl cellulose or the coating pectin. So there's no issue of pore resealing. Hence, the drugs cannot be released because our coating is not really strongly adhered onto the pellet, but it's only a lightly adhered and exerting hydrophobic repulsions to the water medium of the intestinal tract.
Okay, thank you.
Yeah.
Any other questions to Prof. Wang?
I think a very common questions that people will ask is about the microwave. Will they cause any disastrous effect onto the skin? So, and how do I commercialize? We are fighting very hard trying to commercialize, but I foresee there will be a lot of ministerial and administrative hurdles because microwave is not chemical penetration enhancer, it's radiation penetration enhancer. So about the safety, I have done a very simple experiment looking at the lung, liver, and kidney toxicity, a very preliminary study. It doesn't seems to be develop any adverse effect. And I wish to highlight that the microwave that I use is only 1 milliwatt. So unlike those home kitchens microwave, which is 900 to 1500 watt. So ours is only 1 milliwatt. And per cm square of the skin, we expose only 0.14 milliwatt of microwave. So the level of the microwave is very low. And what about long-term use? I foresee no issue because wearing this earphone and switching on my mobile phone, I have received enough microwave, more than 0.14 milliwatt per cm square of my skin. So I'm going to use this to defend against the authority of product registration. Let's see what will happen.
Okay, thank you.
Thank you.
Prof. Wang, having one question from the chat box from Tushar Bate. What vehicle in coating is preferred, solvent versus water?
Solvent versus water.
Water.
When I do coating, I don't use any solvent or any water. So my main water comes from the gastric fluid, come from the intestinal fluid. It can be pure water, it can be water with electrolyte, it can be water with enzymes or water with proteins, or water with a microorganism.
Okay.
So we are doing our...
Sorry, Professor Wong, to jump in between while you are speaking. While you are coating your pallets, you need a vehicle to use or dissolve your Utragit. So that was the reason. What vehicle you used to dissolve that?
I did not use Utragit. Now the example I am giving you just now, Utragit is from other researcher.
So even if you are using say HPMC or ethyl cellulose or any of those coating materials, you would need a vehicle. And I am more concerned on the environment thing. That is why I'm asking you, how do you do that when you are using nanoparticles? And
actually for the colon specific delivery, the coating is effected in vivo, not in vitro. Hence, I do not use any solvent or water to dissolve the coating materials. So basically, you can imagine the coating powder in the form of the solid mixture is blended with the drug pellets in the capsule. And the patient will consume the capsule. And the water from the gastrointestinal tract is the one that elicits the coating.
When you are putting this powder mix into the capsule cell is going to dissolve in less than 30 minutes.
Yes.
And the material will pass and it will not reach the colon because it is already in the acidic medium.
Yes, you are right. The capsule may be disrupted, but fortunately, the pectin is able to act quickly to bind the ethyl cellulose onto the pellets to form a robust plug. We have actually performed the experiment in vitro as well as in vivo now to elucidate the so-called physical structure of the aggregates. So it's still there.
Because professor, if I have to claim that it is going up to the colon, then I have to show that the membrane does not break for 2 hours. That is what the pharmacopoeias usually state. So I don't know how to overcome this challenge.
What membrane is that?
The coating material needs to be stable for at least 2 hours.
Sorry, I think we are actually coming from different direction. So I think in our laboratory, we advocate not to follow the norm. So basically, whatever we do, we try to do something more innovative and trying to resolve the hurdle that the past researcher have encountered. Later on, once the innovative idea is concreted, we will then try to see how to develop into a possibly commercialized item. Now, under such circumstances, we will consider the monograph requirement. Of course, also all the Ministry of Health authorities requests. But at this moment of time, our center actually focus on crazy ideas. In other words, we are not too obedient. We hope to be able to train the student not to be a follower. Now, most of my students likes to follow, right? And so we are trying to train them not to follow and not to be afraid of the teacher. We can argue, we can argue, we can quarrel, but after all, if the student needs to win over me, they needs to read more. So this is a good approach.
Professor, I appreciate your approach. It was just that I come from the industry and I have completed my studies too long back. So it's like I carry an experience of almost 25 years in the industry.
That's good.
I was just trying to understand what ideas you guys are dropping, and being a diabetic patient, I was more interested to know about inhalation. So that was the only reason of jumping in. So those were industry-specific questions on monograph and all those things.
I can share with you our recent research approaches. You can see that I think Yazid is also one of my new postgraduate students. Now, the approach we are taking is very different now. We try to synthesize new polymeric materials, and we encapsulate or we even conjugate the drugs to the polymeric backbone. Now, from the industry perspective, this will need a lot of FDA approval process. Is highly unprofitable. So I think between academia and industry, there's a thin line whereby I think we need to constantly discuss how to work together. So on my part, we have actually found a number of industry partners that are willing to absorb and adopt so-called more difficult to be approved samples for their long-term plan. Now you can see that a lot of industry, even in Malaysia, I think they are not just planning to survive during the lifetime. Now, in the company that I work with, now the son is taking over. So it's just like many India company. You can see that the dad set up, the son or the grandsons take over. So they have a very, how shall I say, long-sighted vision. Hence, certain company, I wouldn't say all, especially company that wish to have a long-term survival over a few hundred or 1,000 years, they will be keen to work with us. And these are the company that we try to be associated with and close to.
Thank you.
Thank you. Hope to meet you in the future.
Sure.
I do visit India twice a year, but unfortunately, this year is okay. Just before COVID, I already visited 2 times. One at Calcutta, and both are Calcutta, but different regions. Yeah.
Okay, thank you very much, Prof. Wang, once again.
Thank you.
So thank you very much for your wonderful presentation.
Thank you.
Now we are moving to second talk. The second speaker for today's webinar is Dr. Sunita Sampati from GITAM School of Pharmacy, Hyderabad, India. She will be delivering a talk on formulation approaches to improve the brain uptake by intranasal delivery. Before the talk, I would like to present a brief introduction of Dr. Sunita Sampati. Dr. Sunita is working as an assistant professor at School of Pharmacy, GITAM University, Hyderabad, India. She was awarded a PhD in Pharmaceutical Sciences from the College of Technology, Usmania University, India in 2011. She is having a total of 18 years of experience in teaching and research. Dr. Sunita's research interest focus on the formulation development using nanosuspension, micro and nanoemulsion, polymeric and lipid nanoparticles, and complexation using QBD approach for antiretroviral, anti-cancer, and anti-osteoporotic drugs, and with a special focus on intranasal delivery for antidepressants. As a principal investigator, she successfully completed 2 government-funded projects. Till today, 30 master and 3 PhD candidates have been graduated, and currently 5 PhD candidates are pursuing their postgraduate studies under her supervision. She has published 42 research and review articles in peer-reviewed international journals with an overall impact of 50. She is also an author for 5 book chapters and is also serving as a reviewer for many pharmaceutical journals. With this brief profile, I now request Dr. Sunita Sampati to start her presentation. Thank you.
Thank you, Dr. Ravi, for your nice introduction. Yeah, I'm going to start my presentation. I'm going to share the presentation. Yeah. Can you see the slides, Dr. Ravi, sir?
Yes, we can see. Yes, ma'am.
Yeah. Once again, a very good afternoon to all the eminent personalities in the field of pharmacy, professors, colleagues, and dear students. I'm here going to share my views on the topic which we have worked out in our labs by our PhD students. This is formulation approaches to improve the brain uptake by intranasal delivery. These are the contents of my presentation. The idea of intranasal drug administration is not completely new, and it has been used in ancient Indian medicine. According to the Charaka Samhita, Nasya is regarded as the best treatment for Shirogas. Acharya Vagbata said, Nasa Sirasodharam means nose is the painless and direct opening for delivery of medicines to the cranial cavity. And one of the ancient Indian medicine which is followed in India is Ayurveda. And in Ayurveda, the treatment is given by Panchakarma, where different types of routes are used for delivering the drugs as well as for cleaning the toxins from the body. And one for such route is the Nasya. So the same nasal route is used by many Ayurvedic drugs also. I'm just presenting a small case study on that. This is the Nasya medication which was given to a coma patient who was 61 years old. She was admitted to hospital for 28 days. Later, the physician has suggested to take the patient to home or continue the physiotherapy. So along with the biomedical approach, the Ayurvedic people, they have even administered 2 types of formulations to her in the form of a nasal powder and in the form of a nasal vapors. So on 7th day, they observed that the patient was able to open the eyes and she was even responding to verbal responses on the 14th day. So though the nasal route of administration may pose challenges to humans, but microbe knows this intranasal route better. And many of the airborne infections are spread via nasal route. So when a traditional system of medicine testifies the use of nasal drug delivery, and many living living organisms in the environment can access this route into the brain easily, how can we utilize this knowledge for drug delivery systems? And brain targeting has always been a challenging to the various, because of the presence of various physiological barriers which are called as blood-brain barriers, and hence resulting in low bioavailability of the drugs when they are administered by conventional strategies like oral or parenteral. And this particular problem we can tackle by delivering the drugs through intranasal route of administration, where the drug reaches to the brain, to all parts of the brain, because of the direct access of these particular medicines to the brain. If you look into the advantages of intranasal delivery, there are many advantages. It is a non-invasive and convenient method of administration. It avoids the drug degradation, which was highly reported when the drugs are given by oral route of administration. We can avoid the first-pass metabolism. We can enhance the bioavailability of the drug. At the same time, when the drugs are given by intranasal delivery, it can circumvent the blood-brain barrier, and we require only a low dose of drugs. There can be a rapid action, and the drug reaches the target site. No need of any modification of the therapy, and dose-related side effects can be reduced. Now, when the drugs are administered by intranasal route, the drug can take any of these particular pathways. So first, it may enter into the olfactory region as well as the respiratory region, and following, it may reach the brain directly or to the CSF. So though intranasal drug delivery system has numerous clinical applications, there is a high incidence of chances that we can use this particular approach for targeting the drugs for the many neurological disorders. If you look into the statistics of neurological disorders, they are increasingly every year, and the deaths related with this particular neurological diseases are also increasing. Hence, we need to have some strategies where we can directly deliver the drugs to these neurological disorders. And many researchers are working in this direction so as to see that the drug reaches to the required target. But as we know that most of the drugs are administered by conventional route of administration, that is either by oral route or the parenteral route. But when the drugs are given by the oral route, especially for the neurological disorders, there are always barriers which are there at the central nervous system. And because of the harsh environmental GI pH, the therapeutic effect of the drug is going to be short, and the drug undergoes first-pass metabolism, thereby reducing the systemic absorption and leading to a poor bioavailability. So therefore, in order to enhance the therapeutic activity, always we increase the dose of the drug and we use it for a long-term therapy. But when they are used for a long-term therapy, there are always side effects related with the dose of the drug. So therefore, we have a number of techniques which can be used for the targeting the central nervous system. And one particular approach is where we can use by means of an alternative routes where the drugs can be administered by intranasal delivery. So there are various carriers which can be used, which are called as nanocarriers. So these are the different types of nanocarriers which can be used, and where the work is highly reported by many researchers for targeting this particular brain by using an intranasal approach. And in our lab, we are focused, we started working in the area of nanosuspensions and micro or nano emulsions for nose to brain targeting delivery. And we are also using solid lipid nanoparticulate approaches, polymeric nanoparticles, as well as nanostructured lipid carriers for other route of administrations. Now, these are the drugs which are there in the clinical trials for nose to brain delivery, and you can see here the oxytocin nasal spray. It is used for the indication of the cognitive and reactive fever, and the status is in the recruiting stage. And these are some of the FDA-approved drugs for intranasal delivery, which are available in the form of a nasal powder, nasal sprays. Now, as I said, we are working in the area of the nanosuspensions. So the question is, do really the nanosuspensions can be used for the CNS via the nose to brain delivery? Are they really going to work? Yes, they are going to work, because The nanoparticles, the nanosuspensions are the one where these are the drugs where it is in the colloidal stage, and the drug is mainly stabilized by using the stabilizers to bring the size to nanometric range. So when the nanosuspension containing the particles less than nanometers, in nanometers definitely they have larger surface area. And the other advantage of the beauty of this nanosuspension technique is that we are using different types of polymers, and some polymers may have the property of adhesiveness, and thereby they retain the particles into the nasal mucosa and increases the solubility of the drugs. Thereby there is an increased rate and amount of the drug that is reaching the site. At the same time, we can expect high concentrations of the drug. And in nanosuspensions, we are not using any toxic solubilizers or co-solvents. We are using the stabilizers as per the inactive ingredient database reported as per the FDA in the limits. Now, there are many approaches for preparing these nanosuspensions. We have a top-down techniques as well as bottom-up approaches. Now, this is the case study which I am going to present, and this is a work which we have done in our laboratory. This is the started work, and we are exploring in this particular area with different formulation approaches. So the first approach what we have taken is a nanosuspension for zolpidem. Zolpidem is the BCS class 2, which is called as biopharmaceutical classification system 2, which has low solubility and high permeability. And the drug is available in the market in the form of a tablet, and the dose of the drug ranges from 75 mg to 150 mg. So when the drug is administered by oral route of administration, the drug undergoes first pass metabolism, leading to low bioavailability of the drug. And at the same time, because of the blood-brain barriers, the entry of the drug into the brain is also limited. Hence, the objective here, what we have planned is that we can increase the solubility of the drug by using a nanosuspension approach, where we have selected two approaches: a sonoprecipitation approach followed with a combination technique with using high pressure homogenizer, and then we have characterized the whatever the formulations we have defined for the particle size by using a Malvern zetasizer as well as scanning electron microscopy, and followed with the ex vivo studies and in vivo studies in the rat models. Now, here we have prepared the nanosuspension by a simple sonoprecipitation technique, where we dissolve the drug in the organic solvent and followed by the addition of the organic solvent in the aqueous stabilizer, and followed by subjecting it to sonation and complete evaporation of the solvent, and then freeze drying the product to get a freeze dried nanosuspension. Now, in the combination technique, as reported by the Lee et al., the combination technique always gives us a low particle size. So hence, after precipitating the drug into the aqueous stabilizer system, we have subjected that to a roto-evaporator to completely evaporate the solvent, and subjected that to high pressure homogenizer at different pressures as well as different cycles. Now, if you look into the results, initially we have selected different solvents which has high solubility for the drug. So depending upon the solubility of the zolpidem, the methanol and acetone were having high solubility. So in the preliminary studies, when acetone and methanol was used for preparing the nanosuspension, the methanol organic solvent or the solvent which was resulting in large particle size which were greater than 2000 and 4000 nanometers. But whereas the acetone was giving us a low particle size. So when compared with the two solvents, so we have further continued only by taking the acetone as an organic solvent. So if you see this particular slide, we have initially selected a single surfactant, and we have used polymeric stabilizers as well as surfactant based stabilizers at a fixed concentration. Except the polymeric stabilizer, which is HPMC, in other cases we can see a larger particle size with a high PDI and the respective zeta potential. But if you look into the HPMC, this is the one which is giving a particle size around 1000 nanometers. Now then we thought even the soya lecithin is giving a very large particle size. So then we have said if these are the polymers and the stabilizers which are available in the lab, so we have selected these things. And we have taken HPMC further and we worked out with the combination of the various surfactant stabilizers. Now when we have worked out in double combination, because combination of stabilizers always gives us a less particle size. So the pluronic F127, which is based on the polypropylene glycols, and HPMC resulted in a particle size of 780 nanometers with a PDI of 0.4 and a zeta potential of 10. But we have seen in the preliminary studies that the soya lecithin is giving a very good zeta potential. So then we thought to combine the pluronics, HPMC and soya lecithin at the fixed concentration, and found that it is giving a particle size of near to 500 but with a very good zeta potential. So hence we have selected the combination of these three stabilizers and worked out in different ratios. We have fixed the ratios of all these stabilizers as per the inactive ingredient database, which is there in the US FDA, and accordingly we have fixed our surfactants concentrations. Now, when we have done the various trials using these particular surfactants in different ratios, we found that a formulation of F18 consisting of a pluronics 127 of 0.3, soya lecithin 0.4 and HPMC 0.3 weight by volume reduced the particle size, or giving a particle size of 514 nanometers on an average, with a low PDI and a very good zeta potential. Hence, we have carried out this particular formulation as an optimized formulation for further studies. Now, whatever the formulation which we have optimized using the sonoprecipitation technique was taken as per combination technique. So, in the combination technique, the optimized formulation, initially we have checked for two process parameters. The first one is the pressure. We varied the pressure from 300 bars to 1500 bars for 3 cycles. And you can see from the graph clearly that when the nanosuspension was prepared at 300 bar, the particle size was around 1400 nanometers. But as the pressure was increased to 1500 bar, the particle size was reduced near to 500 nanometers to 600. So hence we have fixed the pressure to 1500 bar. And later, at 1500 bar, we have continued and increased the number of cycles from 3 to 24. And if you see from the graph, there was a decrease in the particle size at 12 cycles. So that means at 1500 bar, for 12 cycles, the nanosuspension is giving a low particle size of around 350 nanometers. This was measured as per the Malvern Zeta-sizer. And a further increase in the number of cycles did not have any effect on the particle size. So we thought of continuing the high pressure homogenizer at 1500 bar at 12 cycles. Now, this is the particle size and the PDI and zeta potential, which was measured as per the Malvern Zeta-sizer. So where you can see that the particles are having a very good uniformity with a low PDI and a good zeta potential. Later, we have subjected that to scanning electron microscopy. And if you look into the images of the drug, this is a pure drug, as well as the drug which was precipitated from the organic solvent. They are rectangular blocks and where the size was more than 5 microns. The nanosuspension is reported in the C and D, which is prepared with the sonoprecipitation technique. And here you can see the particles are discrete. They are converted to spherical shape. And the particle size with the sonoprecipitation technique was around 200 to 270 nanometers. And all the particles are spherical and individual, they are not agglomerating with each other. And in case of the combination technique, still the particle size has been reduced because of the principle involved in the high pressure homogenization, where it is based in reaction as well as impaction forces involved in the technique. And the particle size here was reduced to 130 to 140 nanometers, and they are spherical and discrete and they are not agglomerating. Now later we have subjected that to stability saturation studies, and we found that the nanosuspensions prepared with the two techniques of sonoprecipitation and combination technique is having high solubility of the drug as compared to the plain drug. In the similar direction, we have then continued for the drug release studies, which we have done in simulated nasal fluids of pH 5.5. So if you look into the graph, the plain drug is releasing less than 15% at 120 minutes. But whereas the nanosuspension prepared with the combination technique was releasing more than near to 80% of the drug is released for 120 minutes, followed by the combination technique, which can be attributed to the reduction in the particle size as explained by Noyes-Whitney equation. later, as per the short-term stability studies which we have done for the nanosuspension, by storing at 2 different temperatures of 5 degrees and 25 degrees, we in fact thought of subjecting this to accelerated stability conditions, but because of the technical problem with the instrument, we couldn't go ahead. And from the data, we can clearly see that the nanosuspension was stable for 1 month with a little increase in the particle size, though it is stable up to even 6 months also, but there was a little increase in the particle size, which may be because of the osteoid ripening. Now, later we have conducted an ex vivo permeability studies using the pore. This is what is called as the nasal mucosa of the pork. So you can see from the graph that in case of the plain drug, less than 20% of the drug was permeated. But if you look into the permeability coefficient, and when the permeability coefficient was calculated, it was very high. For the nanosuspension prepared by the combination technique, it was 0.4489 folds high as compared to the plain drug, which was only 0.1%. So hence we can say that yes, the drug is permeating, and whatever the polymers we are using and the surfactants we are using, they are helping in permeating. One of the surfactants which we have used is soya lecithin, and you know that this particular soya lecithin is an ionic surfactant, it is a liptic surfactant, and it helped in permeating the drug. Now, then we have moved on to the bioanalytical development. Of course, initially we have done all the estimations using UV visible spectrophotometry at the respective landmarks, but to analyze the drug concentrations in the plasma and the brain, we have developed bioanalytical chromatogram in the respective plasma and the brain using the internal standard as olanzapine. So we can clearly distinguish the retention time of the internal standard as well as azotapine. Now, in the pharmacokinetic studies, we have done the studies in male beast rats, where we have used 5 groups, each group consisting of more than 9 animals. And at each time point, we were sacrificing 3 animals and collecting the blood as well as the brain after slight anesthesia under isoflurane. We have sacrificed the animals and collected the brain samples. And the samples, whatever we have collected, they have been homogenized. And the homogenized samples as well as the plasma were extracted with acetonitrile. And in case of the brain samples, we have completely dried the sample and followed by what is called as adding the acetonitrile, so that because the concentrations will be less and we don't want to directly go for that, so therefore we have dried the samples by using the nitrogen. So this is the pharmacokinetic data of the azotapine formulations in plasma as well as in the brain. This is the plasma. So you can see in the plasma, when the drug is given by intravenous route of administration, the concentrations are very high in the first hours as compared to the intranasal. But when we have looked at the concentrations of the brain, the intranasal nanosuspension is showing high concentrations in the brain as compared to the intravenous route. Okay, and accordingly, if you look into the Cmax and the AUCs, the concentrations of the drug in the brain are very high from the nanosuspension, from the respective nanosuspensions. Yeah, and simultaneously we have calculated what is called as drug targeting efficiency and drug transport percentage by subjecting the data or taking the values of the brain intranasal and the plasma intranasal and brain intravenous by the plasma intravenous. So what we found is that the targeting efficiency is very high in case of when the formulation is administered in the form of a nanosuspension. So we can conclude, we can say that, that there is an uptake of the drug. There is an uptake of the drug when the drug is given by intranasal route. Maybe the drug has been taken up by 2 pathways. That is, the first one is what is called as a systemic pathway. So in the systemic pathway, the drug directly reaches the systemic circulation and it hits the brain by transversing the blood-brain barrier. And the second pathway which the drug has taken is the olfactory pathway. So in the olfactory pathway, the drug moves straight into the CSF as well as the brain from the nasal cavity. Hence, it can be assumed that whatever the proportion of the drug is reaching the brain following the intranasal administration, it could be contributed by either of this, both by the systemic pathway as well as olfactory pathway. And whatever the excipients we are using are also playing a major role in showing the high concentrations of the drug. As I said, soya lecithin is increasing the nasal absorption by enhancing the permeability of the drug, whereas the gluconix is known to increase the transcellular transport of the drug when given by nasal cavity, and it also helped in reducing the particle size. And whereas the HPMC, it helped in improving the drug residence time in the nasal cavity because of its mucoadhesive property. So in overall, we can say that yes, nasal route of the administration of the nanosuspension has increased the concentrations of the drug in the brain by 5 times and more, 7 times when the drug is given by a nanosuspension of a sonoprecipitation technique and a combination technique. Now, immediately after these particular studies, we also done the cytotoxicity studies in the various cell lines. So here we have done the cytotoxicity studies in 3 cell lines. The first one is what is called as RPMI cell lines. These are human nasal epithelial cell lines. Or we also call it as a human nasal epithelial cell lines. And the second study, the second cell lines what we used is these are BASE2B cell lines. These 2B cell lines are mainly meant for the human lung epithelial cell lines. And the third cell lines what we have selected is the neuro-2A cell lines. These are the mouse neuroblastoma cell lines. And the reason for selecting all these cell lines is the first RPMI cell lines are generally these are used as the first screening tools for cytotoxicity, as well as for the permeability of the drugs in preclinical evaluation, and where the drugs are given by especially by intranasal route of administration. Because we know that the mucosal layer on the nasal cavity is very sensitive, therefore we need to assess the toxicity of the compounds, whatever we are introducing into the nasal cavity by performing the cell line assay on RPMI cell lines. Then, as we know that when the drug is going to be administered by a nasal route of administration, some amount of the drug also enters into the lung. So therefore, we have checked the cytotoxicity of the other formulation on the bronchial cell lines. And finally, we have also checked the toxicity of the drugs in the neural cell lines, because finally our drug has to reach the brain. So when all these particular cell lines were used and studied for the cell viability, followed by MTT assay where we have calculated the IC50 value, we found that the cytotoxic of all the prepared nanosuspensions as a dose-dependent manner as seen from the cell viability. But if you look into the IC50 values, the bland nanosuspension IC50 values are very high, indicating that the bland nanosuspension containing the excipients are highly safe. And the IC50 of all the other formulations of zodipine plain drug as well as the nano formulations were found to be significantly low and different from that of the blank nanosuspensions. But from this particular study, we can confirm that the nanosuspensions as well as the plain drug did not cause any toxicity at the levels what they have been exposed. Immediately we have, after this particular cytotoxicity studies in the cell lines, we have taken forward for the ex vivo toxicity studies, which is also called as a nasal ciliary toxicity study. So here we have done this particular study in the nasal mucosa of the porcine skin, that is the pig. So we have exposed the formulation to the nasal mucosa of the porcine pig, and then later we have checked for the damage of the ciliary. So the A is a positive control where it has been exposed to the saline, B is a negative control, and as for the literature, the negative control is always maintained by isopropanol, so the same we have used here. And from this particular histopathology section, we found that the negative control where there is a damage of the nasal mucosa, extensive damage has been observed, and there is a loss of nasal cilia also. But the nasal cilia were intact when treated with the blank nanosuspension and followed with our formulation. So from this particular study, we can say that the zodipine formulation in the form of a nanosuspension did not show any harmful effect on nasal mucosa. And this may be attributed or contributed to the pH of the formulation, which is very similar to the pH of the nasal mucosa of 5 to 6.5. And finally, we conclude that the study projected us to come up with a successful formulation of a poorly soluble drug of zodipine as a nanosuspension, and where the optimized formulation is consisting of a combination of 3 surfactants, because as reported by the authors, a combination of surfactants always stabilizes the systems in a better way rather than a single stabilizer. And the nanosuspension, which was prepared by the combination technique, was stable to up to 6 months with a little difference in the particle size. X5 permeability study also enhanced the permeation across the nasal mucosa because of the presence of the various surfactants. And the AUC values of the drug in the brain by both the approaches was approximately 5.6 and 7-fold greater than plain zodipine. Cytotoxicity test of all the formulations on various cell lines proved to be safe. And from the X5 nasal ciliary toxicity study, it found that the formulation did not exhibit any significant differences in the microscopic structures after treating with the zodipine nanosuspension. And finally, we can state, we may say that yes, intranasal zodipine nanosuspension is a better approach to increase the brain distribution of zodipine. So this is our research team, and she is my, all these are my doctorate students who has completed and submitted their thesis and successfully awarded. She is Dr. Sonia, who is working as a postdoc fellow at Ireland. She's Shravanti, who has just completed her PhD, and he's Dr. Nagarjun, who is presently working in a pharmaceutical company. And these are the other students who has contributed and helped Dr. Shravanti. Now I'm just opening for any questions. And before I take up the questions, I thank our principal sir, Professor Shivakumar sir, for motivating me and for giving an opportunity to share my research expertise with the eminent speakers as well as with the audience.
Thank you very much, Dr. Sunita, for your great talk you delivered today. Of course, the presentation is most interesting and highly informative. Thank you once again. And now we are opening the question and answer session.
Ask the first questions.
Yes, please.
Yes, Prof.
You can hear me, yeah?
Yes. Yes.
Hello. Yeah, because now it's raining, so maybe the Wi-Fi is not so good. So, Dr. Sunita, can you rationalize the choice of your carrier materials against what other researchers have adopted?
Dr. Sunita, can you please unmute your mic?
Yeah. Professor Wong, sir, we have selected 3 types of stabilizers here. We have used HPMC as a polymeric stabilizer, and Bluronix F127 and soya lecithin. So these 3 polymers, these 3 surfactants were screened because of their reduced particle size when we were using the sonoprecipitation as an approach. So the reason for selecting HPMC is it is a polymeric stabilizer, and it mainly helps in reducing the polymers, reducing the particle size because of the intermolecular bonding which is causing with the zodipine. Because the HPMC is known to be a hydrogen donor, and zodipine is a hydrogen acceptor. So what we understood is that there is an intermolecular interaction between the drug as well as the selected stabilizers. Soya lecithin has been selected. The reason for selecting the soya lecithin is it is reported as a permeability enhancer when the drugs are given by intranasal route of administration. So hence, we thought of incorporating the soya lecithin as a permeation enhancer, and at the same time, it was also increasing or giving a very good zeta potential. And the other one, what we have selected is the Pluronic F127. This is also a very good stabilizer because it is based upon the polypropylene glycols, and we thought that a combination of the stabilizers are always advantageous for getting a particle size, a low particle size.
So I believe in nano suspension research. Now, many
Sir?
researchers.
Yeah.
Oh, can you hear me? So I believe in nano suspension research. Many researchers have used stabilizer. They may have used the permeation enhancer. So when you select your own choice of material like HPMC and things like that, Fluoronic, what are the advantages that they can confer compared to the previous study done by other researchers? In other words, what are the additional advantages that you attain, but yet other researchers can never reach?
Okay, okay. To be frank, sir, this is the first study which we have taken it for the nanosuspension. Though we have taken the nanosuspension as a route of delivery for oral route, where we work for osteoporosis. Okay, but when we have selected these particular stabilizers for the nasal, the main reason for selecting these things is we were looking into the inactive ingredient database as per the US FDA. So from the side, because when we go with the nasal route as a route of administration, whatever the excipients we select, they need to be highly safe. They should not be toxic. Okay, so that is one of the reason which we have selected these particular excipients. And we have selected these excipients in the range which they are meant to be worked. So in other cases, what we have seen, the researchers are using the excipients, even the range the recommended levels also. But we want to fix ourselves to the range which is recommended by the US FDA. So that is one of the things which we could adapt in our nanosuspension approach.
Okay, thank you. Thank you very much.
Thank you, sir.
Dr. Sunita,
sir.
There is one question from the chat box.
Okay.
One participant, Dr. Vijaya Kuchana, is asking, how did you perform ex vivo permeation?
So coming to the ex vivo permeability studies, from the various articles, what we found is that we can do the ex permeability studies, especially by taking the pork nasal mucosa. So we have done the ex vivo permeability studies in the nasal mucosa of the pork. So we have gone to the slaughterhouse. In fact, my student, we went along and we brought this particular the pork nose and trying to slice mucosa, nasal mucosa, and that we have placed on a France diffusion apparatus. Like what we do in the diffusion studies, we use a France diffusion apparatus, and by placing this particular membrane, we have placed the drug in the vehicle in the acceptor compartment, and we used to collect that samples. And after exposing the drug to the membrane, we have gone for what is called as we have checked how much of the drug has been permeated through this particular nasal mucosa at various time points, and accordingly we have calculated the permeability coefficient. And in case of the nasociliary toxicity study, we have exposed the formulation to the nasal mucosa for respective time periods, and that has been taken. And after what is called as a staining, we have looked into the histopathology of that particular membrane.
Okay, thank you. Any other questions to Dr. Sunita?
I have a question, Dr. Ravi.
Yes, please go ahead.
Good evening. Good evening, everyone. I'm Dr. Zahid from University of Sharjah, and one of the colleague of Dr. Ravi, who is moderator right now. Thank you, Dr. Ravi, for the invitation.
Thank you.
Dr. Sunita, thank you so much for your very interesting and comprehensive presentation, starting from the optimization of your formulation until the toxicity study. So I just want to, I was curious to ask you regarding the optimization.
Okay.
That you have done so many process parameters, you have changed so many formulation parameters to reach to the intended goal, which was the size as well as zeta potential. But when I go through your stability slide, there you mentioned that by increasing the time, the size was increasing. So I just want to ask you, what you think, based on your experimental finding, based on your experience, what do you think what could be the reason for this increase in size? Because it seems a significant increase within 15 days only. So could you please highlight, could you comment on that?
Yes, you're absolutely right. And what was our observation is that, whatever the nano suspensions we kept for the stability, we did not freeze time. In fact, we kept in the form of a suspension itself at the different conditions. That is the first thing. The second thing is maybe it could be related to the ostwald ripening also. Maybe the stabilizers what we were using, especially the HPMC what we were using, maybe it is also resulting into the leading to the ostwald ripening. That could be one of the reasons we can say that there is an increase in particle size. Okay, the same thing we have even checked with our nano suspensions when we were using for osteoporosis also. So there also what we found is that yes, when it is stored, when we have done the freeze drying, the freeze dried product, the stability is more rather than when it is in the form of a suspension. Okay, so the reason why we kept that in the form of a suspension is that during that particular time of our work, our freeze dryer was not completely working, so we could not use the freeze dryer for freeze drying of the particular products. So what we have done is that we have kept the nano suspension as such at different temperatures and checked for the assessed for the particle size as well as for the zeta potential. So maybe one of the reason could be related to the ostwald ripening, according to us.
Thank you, Dr. Sunita. So just one of my suggestion maybe, if you can do like you were doing your stability study and just assessing the size and zeta potential to zeta sizer maybe, I suggest you can also do the scanning electron microscope you have done already in your study. So you can do at different time points. You can also do that study like TEM and SEM, where you will be sure and you will know that what is the reason for increase in size. Either it is an agglomeration of particle or it is an ostwald ripening. So that is my suggestion. Because with the monograph, that will give you some sort of agglomeration if there is, because from the resulting data, I also noticed that the zeta potential, the maximum zeta potential that you achieve is minus 18, I think, minus 18. So what I have experienced mostly, if the zeta potential is lower than minus 30 or plus 30, it has some chances of facilitate aggregation of the particle in later stages. So that's why this was my, so because second, you can also if it is ostwald ripening, you can also try maybe some anti-solvent in suspension that could also be improve the stability of your formulation. So overall, thank you once again for sharing your experience with us. But this one I wanted to just comment or suggest you only. So thank you once again. This is all about my question.
You see, a very good suggestion. Definitely, we will take it up for our next study. And as suggested, yes, you are absolutely right. We will take it up for the stability studies also, the scanning electron microscopy, and as well as for the transmission electron microscopy. Thank you very much.
Thank you, Dr. Jahid.
Secondly, sorry, I'm interrupting again, Dr. Ravi. Sometime ostwald ripening is also because of high PDI. So I mentioned, I just noticed also the PDI was also high, which also might be because of the low pressure that you are using in a high pressure homogenizer. So maybe you can increase slightly the pressure. That will also reduce the PDI and will also avoid the problem of ostwald ripening in your suspension.
Okay, we could use up to the maximum of 1500 bar. So that is the maximum which we can use with the available instrument of the microfluidizer.
Right. So it is not high pressure homogenizer. It was just homogenizer. Am I right?
Yeah.
Okay, because high pressure homogenizer normally can go up to 20,000 or 30,000 even.
Yes,
yeah.
Absolutely.
So thank you once again, Dr. Sunita, and thank you, Dr. Ravi. This is all about what.
Thank you. Dr. Sunita, there is one more question from chat box by BJ Mahendra Kumar. Why only pork mucosa and how its characteristics similar to human being?
Okay. The reason for selecting the pork mucosa is because it is similar to the human itself. When we tried and attempted with the rat, we could unable to collect the mucosa because the rat nose is very small and we could unable to collect that. So hence, when we have gone to the literature, the reports, the references and the literature suggest to whoever doing in this area, they have used the pork mucosa as since it is a little similar to the humans. So that's the reason we could use that. That is one reason. The second reason is that the mucosa surface area of the pork is more as compared to the other animal models. So that is the main reason which we have selected for the studies. The pork mucosa was selected.
Okay. Any other questions to Dr. Sunita?
Good evening, ma'am. This is Subramaniam. Hello.
Yeah, I can hear you. Please go ahead.
Yeah. I'm really happy to be a part of this webinar since the invitation has come from my PhD guide, which I am pursuing from GITAM University, Vizag, from Dr. Surendra Singh's house, sir. And I'm very happy to be a part of this. And I have a question regarding this excipient that you have used, that you are mentioning about, that is the IAG, inactive ingredient substrate.
Yeah.
I hope the pluronics what you have used is supposed to be Polaxamer 407 that you have mentioned, F127, right?
Yeah.
Apart from this 407, F127, you have tried the other P188 also, if I am correct. Did you find any exceptional observation when you were using that particular 188?
As such, no, I can say. Initially, we have screened these particular surfactants only based on the particle size. Only based on the particle size, we have selected. Now, if you look into the data, whatever I have presented, the combination with the Pluronics 188 with the HPMC was little higher when compared to the Pluronics 127. So, definitely the surfactants play a major role, but it also depends upon the technique we take as well as the type of the drug for which we are going to work out. So in our technique, we observe that the pluronic 127 is better when compared to 188. But see, each and every nanosuspension of a drugs are different from drug to drug. So the same stabilizers may not work for the drug which you are going to select also. This is all a trial and error basis which we have to do for the selected and available surfactants, whether it is polymeric or ionic or non-ionic surfactants.
And one more point I would like to know is like that you were mentioning about the inactive ingredients guide that you have used in the formulation. As per their regulations of FDA, I mean US FDA, you are using the same limits for the feasibility of the trial. If suppose you want to be commercialized it in an industry or suppose whatever it is. So what is the maximum quantity that you have used in your trials for the Polaxamer 407 and 188? Can I know the quantity that you have used?
Initially, if you look into the trials, we have screened at 0.3 itself. The individual stabilizers were taken at 0.3. And when we have gone for the combination, there we have fixed the concentration ranges as per the limits. So if you look into that, we have fixed from 0.2 to 0.5, 0.1 to 0.5. That's the range which we have used for the Poloxamer 127. And in case of the soya lecithin, it was up to 0.6, 0.2 to 0.6. So in case of the HPMC, it was up to 0.5.
Okay. Thank you. Thank you very much.
Any other questions, please? If no other questions, I would like to say thank you once again, Dr. Sunita, for your wonderful presentation. And it is very informative and it's very interesting too. Thank you once again.
Thank you, Dr. Ravi. Thank you very much for giving an opportunity.
Thank you. Before we go to closing remarks, a small announcement to the participants. Attendance link is posted in the chat box. Please fill the form for your attendance and valuable feedback. Once you submit the form, e-certificate will be directly lodged into your inbox. So I kindly request all the participants to fill the attendance form. Finally, we come to the concluding part of today's webinar. As Dr. Shariza, Dean, Faculty of Pharmacy, UiTM, is busy with one unexpected meeting, I request Dr. Noor Hayati, Head of Pharmaceutics Department, for the concluding remarks.
Thank you so much, Dr. Ravi. Hi, everyone. My name is Noor Hayati. I'm currently heading Department of Pharmaceutics, Faculty of Pharmacy, UiTM. So at the I'm very sorry. My apology for the hiccups that we experienced at the very beginning of the webinar. Very sorry. So because of our dean is unfortunately not available in the afternoon. So on behalf of the Dean, Faculty of Pharmacy, Associate Professor Dr. Shariza Sahudin, and also on the behalf of our Department of Pharmaceutics and Faculty of Pharmacy as a whole, thank you so much for participating this webinar and making a reality as a collaboration between two different institutions. So I would like to thank GITAM School of Pharmacy. Thank you so much, Prof. Dr. G. Shivakumar, Principal of School of Pharmacy, GITAM. Thank you so much to our speakers, Prof. Dr. Wong Tin Wee from Faculty of Pharmacy, Dr. Sunitha Sampathy, School of Pharmacy, GITAM, thank you. And also to the organizer of the webinar, thank you so much. You guys have done a wonderful work throughout the session. And also to all participants that have spent their time attending this webinar. So we are looking forward for the future collaboration and knowledge sharing in the future. So with that, thank you so much and stay safe, everyone. Thank you.
Thank you very much. Thank you. Have a nice day.
You too.
Thank you.
Thank you very much, Dr. Noor Hayati, for your closing remarks. Now I request Prof. G. Shivakumar, Principal, GITAM School of Pharmacy, Hyderabad, India, for the concluding remarks.
Just to unmute.
Yeah, thank you, Dr. Ravi. It was a wonderful session. The international webinar is made it into a reality. First of all, I would like to thank few people to make this international webinar a successful one and an interesting and an interacting one. So first of all, I would like to thank Dean, Dr. Shariza, UiTM, Faculty of Pharmacy. I would like to also thank the HOD, Dr. Noor Hayati, and then the main person who had been behind the moderation, Dr. Ravi. I would like to extend the thanks. Further, I would like to thank Professor Wong for a wonderful exploration of the extension of his research activities to give an innovative insight to all the participants today. Thanks to Dr. Sunitha, who had given an informative talk in this international webinar. I would also like to extend thanks to our President, Sri Bharat, Vice Chancellor, Professor Shivaramakrishnan, then Pro Vice Chancellor, Sir, of various campuses, Professor N. Shivaprasad, Pro Vice Chancellor, Hyderabad campus. Special thanks to Professor S. Ganapathy, Sir, who is already always instrumental and motivating to us to conduct such webinar sessions, Principal, GITAM Institute of Pharmacy, faculty colleagues who had been directly and indirectly helping through this particular international webinar session. Thanks to IT team who had supported us a lot in spite of initial hiccups. Hiccups are always a better learning session for all of us. Thanks to all the participants who have interacted and then made the session very successful. Thanks to all the faculty Thanks to the public and media cell. Okay. Thanks one and all. In fact, we would be looking forward to the UiTM faculty members to have a potential collaboration in terms of research in the future days ahead. Thank you one and all. Stay safe. Thank you.
Thank you very much, Prof. Shivakumar, for your closing remarks. Thank you very much for your participation in today's webinar. And once again, I request all the participants to fill the attendance form posted in the chat box. E-certificate will be provided to the participants through email once you submit the form. With this, we come to the closing of the international webinar with a strong belief that all the participants are benefited with the updated information on the current challenges, recent emerging trends, and innovative technologies in the drug delivery for the tailored treatment of chronic diseases. Thank you once again, all the participants and also the speakers. And thank you once again. Be safe and take care and see you once again. Thank you.
Thank you.
Thanks a lot.
Be safe. Happy day.
Thank you.
Thank you.
Thank you.